Hello, everyone. Welcome to our webinar. Uh, Today, we are going to be talking about how a drug can be crowdsourced and is this myth or reality. And today we will introduce you to a story of collaboration between two great companies, Enamine and PostEra. And discuss some of the technologies that we are using together to have a rapid drug discovery. Uh, Enamine, this webinar is brought to you by Enamine, which is a global supplier of building blocks and compound libraries for drug discovery. We have been serving the industry since 1991. And Enamine is the creator of a real chemical space, which now includes 13 billion molecules and growing. PostEra offers machine learning guided medicinal chemistry as a service. The company combines the world's largest chemical databases with its synthetic technology to get compounds really fast. Today, we will have a number of presenters from two companies. And so let me introduce everyone. Dr. Tatiana Matvyiuk is the director of library synthesis at Enamine. She's an experienced researcher with deep expertise in computational drug design, and she holds PhD in organic chemistry. Dr. Yurii Moroz is leading the real chemical space project at Enamine. He's the leading scientist and PhD in organic chemistry. And myself, I have PhD in physical chemistry, and I'm leading marketing efforts at Enamine, and also doing some market research for the pharmaceutical industry. Our PostEra guest today is Yurii Moroz. He's co-founder and CEO at PostEra. Aaron is a mathematician by training, and he used applied mathematics to solve a lot of complex tasks during his career. But now he spent 5 years in finance as a data scientist and a trader, and then he went into biotech and decided to use his knowledge in the computation to actually launch the PostEra company. Dr. Alpha Lee is a co-founder of PostEra. He's a faculty member at the University of Cambridge, and he's Forbes 30 under 30 winner. And today we also have Dr. Emily Ripka. She's the full stack developer at PostEra, and she's responsible for software development and elegant solutions that the company brings to its customers. Emily holds PhD in physical chemistry as well. Today we will be discussing a number of topics over a course of approximately 2 hours. I will introduce basically I already introduced the webinar, and then Dr. Alpha Lee with Tatiana Matvyiuk will discuss the collaboration between Enamine and PostEra and the COVID Moonshot project, which is a very bold initiative, open science initiative directed to rapidly discovering a drug against coronavirus. Then Aaron will introduce AI-driven synthesis planning approach developed by PostEra and speak about the new product that they have built, the Manifold software package. And then Dr. Emily will basically introduce the Manifold in her demo. Dr. Yurii Moroz then will guide you through the concept and practical application of real approach to ultra large chemical space that Enamine is pioneering. And finally, I will introduce you to a little bit general ideas about how AI is used for drug design by the leading companies. What are the trends and what's going on in this space today? Then we will be happy to answer some of your questions, and then I will finalize the webinar. So with that, I'm passing the floor to our first presenter, Dr. Alpha Lee. And Alpha Lee, welcome.
Yes, thank you very much for the kind introduction, and pleasure meeting all of you virtually. In my talk today, I would discuss the COVID Moonshot Consortium and our initiative, and walk you through our story so far, our progress, and where we want to be, and discuss crowdsourced drug discovery as a concept and how our AI technology has helped in this effort. And we are obviously really delighted to collaborate with valued partners like Enamine, who has been driving the synthesis process in this project. I would like to first take a step back and maybe ask a provocative question of what is the goal of COVID Moonshot, more generally. So what is missing in the picture in our strategies against COVID? Seeing we heard a lot about vaccines and other therapeutic interventions, and I think what COVID Moonshot tries to develop, our goal is a widely available an oral antiviral that can directly target SARS-CoV-2. And that's really important because there's a large window of opportunity actually for antivirals to work. And that's when patients develop mild to moderate illness. And it is known that actually the end stage COVID is not caused by viral infection, but caused by immune response. But the early stage COVID can be aptly targeted in principle by antivirals. And that gives a large window opportunity for either pre-exposure prophylactic, taking antivirals as a precautionary measure just after one has been exposed to patients, or shortly after developing mild to moderate illness. And antivirals are complementary to vaccines and public health interventions, seeing as vaccines are, as we all know, not 100% effective, and there's still a hurdle in distribution and uptake. And more importantly, an orally available pill is just much easier to make, distribute, and administer to patients. And that's why we focus on an oral antiviral as the goal of Moonshot. Within this space of oral antiviral, there are many targets one can choose from, and Moonshot focuses on a validated target called the main protease. The main protease is responsible in a key stage of viral replication. Therefore, inhibiting the main protease would stop the virus from replicating and therefore achieve its therapeutic effect. Now, there is no main protease inhibitor against human coronavirus on the market. One is undergoing clinical trial by Pfizer, but that's an IV drug, not an oral drug. And Moonshot aims to develop the first oral antiviral against the main protease. And the challenge, and again, we picked the main protease because it is both an important part of the viral replication mechanism in COVID-19, but also because it is broadly conserved across different coronaviruses. Here we can see sequence alignments with SARS-CoV-2, SARS-CoV, MERS, etc. And the main protease is broadly conserved, which means that an antiviral drug against COVID-19 would not only be an effective strategy against the pandemic, but also give us a way out for pandemic preparedness in the future. And all drugs and all candidates, which has been floated around in the literature, and in the case of one particular candidate undergoing clinical trial right now, are a class of molecules known as peptidomimetic. Those are large molecules that are peptide-like and therefore often difficult to develop into oral therapeutic. Most actually not only are peptide-like, but also covalent. It means that it binds to the main protease via an irreversible mechanism, and that often incurs downstream risk. And the premise of Moonshot is to get to a potent small molecule, non-covalent, non-peptidomimetic molecule as quickly as possible. And Moonshot, the whole idea of Moonshot builds upon the scientific work done at Oxford Diamond and the Weizmann. Back in February 2020, the main protease was cloned and produced. Frank and his group determined the atomic structure of the main protease, and then Nia and Frank did a covalent fragment screen that shows a beautiful constellation of fragments populating the binding site. And in March, they released 1,500 crystals all in a day. And at that point, the PostEra team back when California was actually looking at Twitter feed, and we realized that, hey, there's like a large constellation of fragments covering the active site. Here are a few zoomed in figures of Frank's original work showing beautiful and densely populated binding site. And all the data is actually available. So we thought, why not launch a crowdsourcing initiative, the Moonshot platform, that asks basically chemists around the world to look at these fragment hits and ideate how could these fragments be merged or expanded into active compounds. And so we quickly assembled a website and a platform to solicit a crowdsourced contribution. Again, asking our communities around the world. Back then, it was, I think most of the world was still working from home and under lockdown around a year prior. And we asked them, what would be your ideas given these fragment hits? We expected perhaps, maybe 100 submissions. It is an acquired taste looking at fragments. And we were extremely humbled and pleasantly surprised that we actually got now today 16,000 compounds submitted to this crowdsourcing platform with 450 colleagues, designers around the world working with us, thinking about what designs to make. And now we have synthesized and tested, thanks to the amazing efforts at Enamine, 1,700 compounds. And I will discuss in the next few slides the chemical progress from hit finding to hit to lead in this program. And what enables, part of the reason why we were able to move particularly rapidly is the technology that PostEra has been developing and has continued to develop, which is the part that Aaron will talk about, known as Manifold. Manifold allows us to quickly analyze and triage compounds based on synthetic accessibility. For example, this one of the lead compounds shown in the left, we found out is actually a one-step synthesis from Enamine in-stock building block. And Manifold is a technology powered by artificial intelligence and machine learning, which allows us to quickly triage synthetic accessibility of compounds and rapidly come up with retrosynthetic routes. And in the first week of Moonshot, we solicited over 2,000 submissions, and Manifold went through all thousands of submissions within a day or two and picked out which compounds can be easily made. And that really accelerates the hit finding and subsequent lead optimization progress by giving us a very concrete understanding of chemistry challenges. And Moonshot has blossomed into a global collaboration with trusted partners like Enamine, but also folks at MSKCC doing FEP calculations with the Folding at Home Consortium, and also many pharma collaborators, for example, UCB Pharma, and many others, Magchemica as small biotech in the UK, and many other partners around the world offering in-kind support. And importantly, all of our data is released immediately online. It's a completely open science approach where none of us take any patent rights to data. And we really hope that the data seeded a community of open source drug discovery and in general, trying to understand coronavirus and discover therapeutics against it. So a few slides to summarize where we are, our journey towards the lead compound. The compound on the far left was a crowdsourced compound from the fragment from the fragmented Diamond and Weitzman and others released. Almost week one, a member of the community suggested this one-step synthesis from Enamine building block, which model picked up. And then that was swiftly built upon by the community when we released the data back. And now we are at an 80 nanomolar lead compound with nanomolar antiviral activity against COVID-19. Note that these are extremely small molecules. They are very ligand efficient. unlike a peptidomimetic, and they are also non-covalent. So they are really small ligand efficient and non-covalent molecules exhibiting nanomolar affinity to the main protease and nanomolar inhibition against the live virus. We also were able to solve a lot of ADME issues. I talked about potency in the last slide. Our original lead compound shown in the middle has solubility and clearance issues, and we are able to rapidly in a synthetically accessible way solve these ADME and solubility issues. Now our lead compound is reasonably good solubility, has reasonably good solubility as well as satisfactory clearance properties. And our lead compounds are now undergoing PK studies to further characterize its pharmacokinetics properties. What we've also done during Moonshot is to build on not only synthesis technology at PostEra, but also our generative technology to generate new scaffolds and hop in scaffold space with machine learning. And here's an example of what we have done to help the compound generation process. These are on the left are 3 compounds that are the top scoring compounds in July, and we applied the PostEra algorithm to generate new chemical scaffolds, and actually we found compounds that are potent and exhibit good antiviral properties, and that just opens up the scaffold space for further exploration. And I think we have been, and I think the synergy of crowdsource approach providing creativity and the machine learning approach of giving us ideas about synthesis and also giving us alternative exploration of scaffold space allows the program to move in a very rapid way, achieving a nanomolar compound from fragments in less than a year. And in terms of where we want to be as a program, this is our target product profile and where we are right now. We want the compound to exhibit IC50 less than 50 nanomolar. Now we're at 80 nanomolar. We want viral replication to be around 200 nanomolar. We already had around 500 nanomolar, so we're very close. And the PK studies are currently being done right now. And solubility again is something we have achieved. And safety profile, we have profiled our compound against a broad protease panel, and it's clean against that. We also did a safety pharmacology panel, and the lead compound is also clean against that panel. So in summary, we think we are a few months from declaring a preclinical candidate, which will be ready for IND enabling studies. And again, I think our approach of starting from fragments and sort of ideating novel scaffolds through combination of crowdsourcing, machine learning based synthesis prediction, and generative model allows us to sort of break away from a lot of the old chemotypes and going into really ligand efficient small molecules with favorable biological profile. This is a little bit of a preview. We not only have a lead compound, but now a leaderboard, a set of compounds which are favorable. Those are really small compounds, again, highly ligand efficient, showing good antiviral activity, good ME properties, minimal off-target liabilities, and PK properties are now being characterized as we speak. So to summarize, Moonshot is rapidly progressing towards an oral antiviral preclinical candidate against COVID-19. But beyond our lead compounds, I think Moonshot has yielded several benefits. First is a rich source of main protease data, both structural data. The Oxford Diamond Group released over 300 ligand-bound MPRO structures, which I think would be really important to the field, not only for this project, but also to understand about physics of MPRO and also of future pandemics. We have also nucleated an open science infrastructure to coordinate drug discovery. And I hope also showed that open science, a collaborative approach to drug discovery, does work in pushing a rapidly moving drug discovery campaign. And in the process of helping drive Moonshot, PostEra also built an AI-powered synthesis technology, the Manifold platform, which Aaron and Emily will talk about later on in this symposium. And on that note, I would like to hand over to our collaborator, Tatiana, who will be telling us a little bit more about what Enamine is, a very powerful chemistry resources.
Thank you, Alpha, for your kind and detailed explanation of the project. I think that should be now clear in general for our listeners. And I would like to give a short, like general introduction on Enamine company, and also and also on our part in this project. Overall. Okay, I will share my screen. A minute. So Enamine is scientifically driven, privately owned chemical company with over 30 years experience in this field. All our research and production facilities are located in Ukraine, in Kiev. So for the moment, we have over 650 chemists, synthetic chemists, and with 60 PhD levels chemists. So we have no own drug research, and Enamine is entirely a service-oriented company. Now, with a drastic evolution for the last, I would say, 5 years. In total, we have like a half and a half distribution between our business, like in a chemistry catalog business, and as a CRO company with a service provider. We have, Enamine has the largest building block, the world's largest building block collection, and also the largest screening collection, which gives to our customers unprecedented opportunity for fast follow-up, like in libraries, both in library synthesis and analogs, search of analogs from in-stock collection. Also within standard custom chemistry services, we offer medicinal chemistry support with some computational chemistry support, and also we have well-established biology service laboratory on board with HDS opportunity, ADMET, and in vivo PK studies. Enamine also actively participate in a higher education process. We have like a common PhD programs with other European and US institutions. Also, we collaborate with a leading academic institution. And also our synthetic chemists, they like to work on development of new hematypes and new scaffolds. So that's give us an opportunity to issue an over 50 scientific papers per year. So here's just a general scheme, how Enamine support drug discovery. So that's just a general slide which represent that except the largest screening collection and building block collection, we have also fully equipped biology services on board and in the same place. Within this unprecedented crowdsourcing project, and we are happy to collaborate with PostEra, which established quite good communication and holding all the structures for all this project. As I told, so at the beginning on the March, as Alpha already mentioned, everything started from a diamond light source where Frank von Delft with his research group collected a number of X-rays for the first X-ray structure of main protease. Then together with Alpha, they call it to Enamine. They write the letter if Enamine would like to participate in this crowdsources open project. For us, it was really interesting and a chance to see all drug discovery process. So Enamine and this process is responsible for the synthesis, logistic, and compound management. So for the moment, we have 10 FTE chemists, and we are doing we are doing singleton synthesis, lead compound, lead series, and also we are synthesizing doing heat follow-up libraries. Using our automated parallel chemistry laboratory, which give us an opportunity to synthesize up to 2,000 molecules per month. Also within the project, we are doing all the compound formatting and logistics. So each week we have shipments with the newly synthesized compounds into 4 institutions, to the 4 institutions. To the Weizmann Institute of Science, where the group of near London developed extremely fast and robust method of enzymatic MPRO screening, and they give us an update and results very quickly, just a few days after they receive compounds. We have all the activity data with the AC50 curves and other things. Also, another plate with weekly shipment goes to the diamond light source, where at XChem group, leading by Frank von Delft, they are doing X-ray screening of newly synthesized compounds, and also in the same time, just within a week or 2, we can have an X-ray structure of the most active compounds, which give unprecedented opportunity for quick follow-up. Another plate to go to the University of Oxford, to the Scaffold group, where they doing rapid fire mass spec screening with the rapid fire. And another plate goes to the University of Chicago for viral assay. So also, Enamine is responsible for the compound storage and management. So we are doing the different formatting and sending all the selected compounds to the different institutions all over the world for viral assay, PK studies, and all other investigation. Thanks to a very flexible communication, weekly communication with Medchem group and Moonshot project core team, we action very quickly on any following stage. And the decision-making process is quite short for such large projects. Also, we other scientific groups and commercial collaborators, which Alpha mentioned before, are sending compounds to us for the general formatting and store and logistic and further logistic, and also the like reference compound and some other interesting compounds came from the other commercial vendors. And all these all Moonshot compounds are kept and storage at Enamine. Also, we use our on-site biology service. So, for example, within the Moonshot project, the number of biological studies like mouse PK, human liver microsome stability, thermodynamic solubility, and other have been used for the evaluation of safety of compounds and unmet profile. All this also make shorter time needed for the discovery of new lead series and focusing on the right compounds, which we can proceed further with a good profile. Also, to summarize what was done for less than 1 year. Here are some general notes. So we synthesized more than about 1,700 compounds. About 10 heat follow-up libraries have been synthesized. The goods were obtained also thanks to the great medicinal chemistry group, which with a good experience in pharma. And also we have the 3 lead series have been identified within the project and with a focus on the promising one, which Alpha mentioned before. Okay, so thank you for your attention, and I will be glad to answer for any of your questions.
Brilliant. Thank you so much, Tatiana. As Andrei mentioned at the beginning, I'm just going to spend kind of like 10 minutes here just giving an overview of the synthesis technology that Alpha referred to. It's an incredibly powerful platform that really helped Moonshot and that we've now made available to all of you. So just a particular thanks again for everyone who's taken the time to attend this webinar. So I'm just going to share a few slides with you before handing over to Emily, who's going to give you a demo of the platform. Just for anyone who is not aware of PostEra as a company, so in essence, we really focus squarely on medicinal chemistry. We use the latest advances in machine learning that our team pioneers to really help drug discovery initiatives. They're not all open source, but Moonshot happens to be. And we really have developed a suite of tools that we believe can really drastically reduce the cycle times of the clients that we collaborate with. We're very proud as an organization that our technology is not a black box. Machine learning has come under a lot of criticism for that. You are very free to read the publications that underpin PostEra's technology. This technology has been validated with some of the largest pharma companies in the world and published in some of the leading peer-reviewed journals. And so I hope it really gives a good foundation of confidence for the application of PostEra's technology to drug discovery campaigns. We work with organizations in 2 different ways. The first way that we work with organizations is via partnerships. So this is where we come on board and we collaborate with you on a preclinical, typically small molecule drug discovery program. We've also done collaborations where we center actually on innovating on machine learning technology, and you can see some of the clients that we've worked with there. Specifically, we actually also take our technology and offer what may be called synthesis as a service, which is where we work with smaller biotechs, two really great companies at the bottom there, where we basically help with the kind of hit expansion and rapid synthesis cycles that effectively really characterized the COVID Moonshot effort. So in some way, PostEra has an end-to-end medicinal chemistry platform driven by machine learning, and we take a slice of that technology platform, which is called Manifold, that pertains to synthesis. So just quickly, I want to come back to COVID Moonshot and just pick up on one thing that Alpha didn't yet get the chance to mention. And that was actually some of the problems that we ran into, some of the pain points that really initiated the need to build a solution such as Manifold. And well, the first kind of challenge we came across was that as we welcomed all of these submissions, there was a very clear delineation between which compounds could be purchased and which compounds needed to go into the synthesis queue. We actually, as Alpha mentioned, work with several of the world's largest pharma, and we had a contributor from one of these companies who had generously donated a lot of their time and work and come up with 16 compounds that they specifically allocated to the synthesis queue because according to their internal tools, you couldn't purchase them. And it actually turned out that 9 of those 16 compounds that were submitted for synthesis could in fact be bought. And I think this really began to signal to us that even the largest, most established companies may not necessarily have the available tools to search the incredibly large virtual spaces that exist. And even if those tools do exist, often they can become a little outdated in terms of the inventory that they have access to. Often they can be a little disparate, spread across different departments. And so what we wanted to do was bring all of this under one house and one solution. We were also at the start realizing that our cycle times were being slowed down. We were working with brilliant partners like Enamine and other CROs. It would have been really helpful to have a solid track of which CROs have what inventory. And so again, PostEra now has a dynamically updated inventory of the CROs like Enamine, and so therefore allowing us to really allocate synthesis to the most appropriate providers. Finally, often it was very clear that people who were submitting ideas probably had very little idea actually how synthetically accessible the actual compounds they were designing were. And really, again, that drove a necessity for having a really high throughput synthesis triaging. And so these were some of the problems that really drove the decision to build Manifold. And so in a nutshell, Manifold now offers synthesis and search across every available molecule. And really it answers some of the 3 kind of key challenge points that we face, which is how do we have a dynamically updated inventory of well over 10 billion purchasable molecules that can now be purchased straight off the internet. It's been a huge engineering effort to be able to associate and integrate all of those, but I think that's just so important because we take a very pragmatic view of doing synthesis. We think particularly at the early stages of preclinical drug discovery, whether you are just expanding around your hits or going from hit to lead, that really doing 4, 5, 6-step synthesis at that early stage is often not the most optimal approach for designing new compounds. It really comes down to, can you do fast, rapid, reliable chemistry in order to quickly search the design hypothesis that you're trying to test and analyze the chemical space around your hits? And so that is really in the second point what the Manifold platform does. It can be viewed in a way as a hit expansion platform, whether your hits are coming from DNA encoded library screening, high throughput screening, or fragment screening, as in the case of Moonshot. What Emily is going to demo to you is how you can quickly do hit expansion inspired by synthesis. And the final solution that we really offer is library synthesis. This is something that we are incredibly fond of and we use an awful lot within PostEra, and the ability to leverage synthetic intermediates to rapidly access chemical space, and to be able to quickly generate a lot of designs from a few key intermediates, again, really drives down the cycle time and allows you to move very, very quickly. So just a couple of key features that I'd like to kind of point out to you about Manifold. In the area of synthesis, every single synthetic route that Manifold designs for you will always disconnect back to purchasable compounds. You will not have to go searching across the internet to try and find out if these building blocks can be made. Secondly, we actually prioritize the route both by synthetic success and also by the strategic diversity of the bonds that have been broken and formed, in giving you a wide variety of options in terms of the synthesis. This means you won't have to crawl through hundreds of potential options and figure out which one is actually viable for you. And finally, the huge engineering effort of keeping the chemical databases that are provided by great organizations like Enamine, keeping that updated and integrating that into the retrosynthesis is something that, well, again, is freely available to you through Manifold. And I think this really speaks again to where we see the advantage of new technologies like machine learning. There's often a very interesting debate in the field as to whether AI or a chemist can predict the outcome of a chemical reaction better than the other. We think that's a very interesting debate, but what we know categorically is that a machine will always outperform a human when it comes to memorizing millions and even billions of available building blocks and keeping that inventory updated in one's mind. So we really like to play to the strength of computation while regaining the benefits that chemists have. Search and similarity is, again, something that became incredibly important in Moonshot. Within Manifold, we offer similarity and substructure search, of which we offer 3 flavors across well over 1 billion compounds, often in under a second, and actually exact search for your compounds across over 10 billion available molecules. I think there certainly can potentially be a view that isn't similarity in search, isn't this a solved problem? Isn't this something that was fixed 10, 15 years ago? And again, what we find is that though those tools do exist in a form, often they are disparate, spread across various pieces of software. They certainly don't often have the updated inventory coming from the increasingly large virtual spaces. And so again, we just want to make it really easy and straightforward and fast for you to be able to benefit from the search and similarity that we offer. And finally, we actually realized that for many organizations that ordering compounds can actually take up some time. We offer through PostEra a very easy way for you to order compounds. We obviously spend most of our week talking to different vendors and partners like Enamine. And so particularly for small biotechs who I know this can be a logistical overhead, we make it very easy for you to just see through Manifold the routes that can be made, and then allow PostEra to work with Enamine and other vendors to just get the compounds synthesized and deliver them straight back to your assay. Now, there obviously are other, in terms of synthesis, other tools out there, and PostEra has done, some level of benchmarking to give you a confidence in the underlying artificial intelligence that we're using. So here is a kind of straight head-to-head comparison against 2 other pieces of open source software. You can see that Manifold tends to find many more routes and does it much, much faster. And I think, again, what I'd comment here is that some of this may well be to the fact that the actual artificial intelligence model is the state of the art, which we believe it is. But in addition to that, often you just find more routes because you have far more starting material. And that just, again, speaks to the value of having a really large, comprehensive underlying inventory to the actual retrosynthesis that you are wanting to find out. And so finally, how can you engage with Manifold? Well, there are 2 different variations. The first is Manifold Pro. So this is all of our synthesis, search, and similarity that Emily is about to demo to you. We offer anybody a free 30-day trial with no commitment. We don't ask for your credit card. And if you are working full-time in academia, we will provide you a free license to use perpetually for Manifold Pro. So please do get in touch if that is of interest to you. We also add a additional kind of a feature of Manifold, which is where we can actually deploy Manifold via a private cloud specific just to you, that is separate even from PostEra's cloud infrastructure. In addition, through this enterprise version, we can actually customize the underlying inventory. So if you have in-house compounds that you would like to use, or whether you have certain suppliers that you prefer to work with, through the enterprise model, we can customize that specifically to you. And there's a big gold padlock there because we recognize how important security is for so many organizations, certainly when you're entering chemical structures into software. And I really would like to give people confidence that the utmost caution and prudence has been put into the development of the Manifold platform. The entire infrastructure is HIPAA eligible from start to finish. We never retain any of the structures that you enter into Manifold unless you specifically ask us to save them. Enterprise takes this even one step further in basically carving out your own private cloud deployment. So again, we really have gone to the extra lengths to make this comfortable and assuring for everyone. This is again just a quick summary of the various features that come both from Manifold Pro and also from the enterprise version. And so in summary, PostEra has really innovated across the entire medicinal chemistry workflow. We would like to really admonish and thank so many different partners that have worked with us on Moonshot, where PostEra's medicinal chemistry platform has worked hand in hand with kind of leading medicinal chemists at top pharmaceuticals and biotechs around the world. And so if that is of interest, a partnership across PostEra's technology would be delighted to do that. But again, we just want to hone in specifically on Manifold today that really pertains to synthesis and search that often can be most useful in the hit to lead stage. If you have any questions at all about partnerships with PostEra or Manifold, you can always reach out to us at that email address at the bottom. But with that, I'm going to hand over to Emily, who just for the next 10-15 minutes is going to give you a overview and a demo of Manifold.
Great, thanks Aaron so much. So yeah, I'm just going to take a little bit of your time here and let's break out of the webinar slides and I'll kind of just get my hands in and show you some of the points that Aaron and Alpha have mentioned about Manifold in real time. So I'm just going to go ahead and share my screen over here. All right, so what we've got going on over here is just, I'm at postera.ai. And so if you just go ahead and Google that guy, it'll be brought to this website here. And I'm just going to focus on Manifold today. So you can just click on the Manifold button up there. And essentially what you're seeing here at this first landing page is just our way to start up a search query. And you can see there's a lot of different ways to enter in your molecules. So you can draw your smiles, or you can draw your molecules, you can enter them as smiles, upload several smiles in a list or a file of SDF file or a bunch of smiles. And you can also do some smart searching if that's of interest to you. So I just have a couple of case studies, which I'm going to use to just demo a lot of the features and really just talk through the different things which we've implemented into Manifold and how they can be helpful for that hit lead and lead expansion. So I'm just going to load in this first example here. And this molecule here was actually one of our early stage Moonshot compounds. And it's actually just a nice molecule to demonstrate some of the features of Manifold. So you can load up a query with a lot of different molecules. I'm just going to focus on this one for now, but you can go up to 25. So I'm going to go ahead and press search. And when you search the molecule in Manifold, it'll bring you to this initial landing page here. And I know there's a lot going on, but we'll kind of just dive through each bit individually and explain what everything can do to help you out. So I'm just going to kind of just filter things down to Enamine because that's just for the sake of this demo here. So if you can see up top, we've got 5 different tabs. So these are just the main 5 offerings that Manifold will display to you for a synthetic search. So the first are the synthetic routes, and I'll come back to these guys in a little bit with another example. But as you can see, we have 12 routes suggested to you, which can be synthesized with a 6-week lead time, all from Enamine. And you can just scroll through quick and see that we've got a lot of 1-step and 2-step routes here. So what I'll do now is I'll kind of scroll through each one of these tabs. So the next one over is the exact search tab. There's no matches here, but almost instantaneously we search over 10 billion molecules, building blocks over Enamine Real, Wuxi Galaxy, and MQ Ultimate. So just a lot of molecules you can investigate here for exact purchase of the molecule, and also across public databases of PubChem and SureChem if there was any previous literature references. If you go a tab over, we've got the similarity search. And so this is ordered by Tanimoto similarity. And you can see there's over 100 matches here for the similarity search. It's going to make this a little bigger, I think, here. And so what you can see is that if you scroll over any single one of these molecules, they kind of light up. And what that's showing you is that if you click on any molecule on Manifold anywhere, it will open up another tab with another search query. So you can just like traverse through all of the similar and like substructure matches, as well as when you're investigating through synthetic routes. So I'm just going to close this one out and go back to the example I was showing you guys. Another thing I'll point out is that on each one of these molecule cards, you have a vendor ID button right here. And if you click on any of these vendor ID buttons, what it'll do is it'll bring you to that compound. So this is an Enamine Real compound there. And so there also will be Enamine like in stock molecules, which are purchasable, and you can follow those links to just go ahead and purchase them. The next tab over is the substructure search. And as Aaron was saying, we've got 3 different flavors of substructure. The first just being superstructures, where your query molecule is contained in that molecule. We also have 2 different kinds of Merkle scaffold, which account for or don't account for the position of the side chains. And so you can see that we've got 9 and 8 respectively for matches there. And you can also see that while some are purchasable from Enamine, a couple of these are just literature references. So these are just the database, the patent databases here. So the last main tab up here on the Manifold landing page are chemical properties and alerts. So if you look here, we've got a little table of some chemical properties and as well as some medicinal chemistry alerts. So these are from literature as well as industry standards like Payne's filters, GSK, Bristol-Myers Squibb. And so this is just here for your like triaging, so you can quickly kind of rule out any screening compounds. So one other thing I'll mention here is if you jump back, you probably saw this export button on a couple different locations. And so what this export button is for is so that if you have a set of substructures or similarity searches which you wanted to get the vendor information as well as the properties in bulk, you can go ahead and you can export there, and you can download a CSV or an SDF. I'll describe the CSV file quick. And so if I just pull it up in Excel over here, you can see that all of these chemical properties, the smiles and chihis, as well as the vendor ID numbers are all here for you to use for the properties, so you can use them in your like maybe external tools for like further triaging, or you could just go ahead and purchase the molecules from the vendor if you wanted to. So I'll come back to Manifold here. And as Aaron was mentioning, we don't by default save any of your search queries. So if you search a sensitive smile string, and no need to worry because this isn't saved. It's here for you to view and explore, but then it's gone. You can go ahead and save it if you really wanted to and come back and explore later on. You could save this query and it'll turn green there. And then in your account, you have like some saved searches if you wanted to, and you can go ahead and delete those guys at any time. But the next thing I'm going to do is pull up another example, kind of a little toy example, which I think is an interesting illustration of the power of Manifold. So when we're reading in literature, we always look for examples which we think would be interesting to kind of explore using the tools that Manifold has to offer. And one in particular, which we found in the literature, was this scheme over here. So if you look over to the right, just no references or anything, no pointing fingers or anything, but this scheme over here, you can see that the authors were trying to synthesize these 4 molecules, and they're actually use 2 and 3-step approaches here. And whenever we see an example like this, it really, we can't help ourselves but to kind of plug these into Manifold to see if not only these are all purchasable, but if we can suggest synthetic routes which are maybe simpler or even the same that the chemists were suggesting. So I just have all 4 of these smiles already loaded up in a file here. So again, you can upload a file with just a bunch of smile strings or some SDF files, and will automatically populate the search query with all of those molecules. So we've got 4 loaded up here, and these are just the A, B, C, and D products over here on the right. So I might just scooch that down a little bit. So if you go ahead and search, you'll be brought to this landing page, which you haven't seen yet because we just used 1 molecule. But what's going on here is that this is just the search results for all 4 of these search queries. And what's really nice is that it shows you off the bat that all 4 are actually purchasable from several vendors. So all of them from Enamine, and also from like eMolecules, MQL, as well as some database references. And honestly, your search could end here. You could go ahead and you could export here, and you could get that CSV, which has all of the properties for these guys as well, chemical properties, the alerts, as well as all of the vendor IDs. So, I mean, right there, you can save a lot of human power and time just by purchasing these instead of going through all of the synthesis to make these 4 molecules to just investigate these like substituents. However, I'm going to go a step further too, and I'm going to click on one of these guys, and I want to see the synthetic routes that Manifold kind of delivered to see how they compare to the chemist's proposed synthesis here. So I'm going to click on number 3 here, which corresponds to C in the scheme on the right. So if we go ahead and click on that guy, our retrosynthesis algorithms, which again, they're state-of-the-art molecular transformer, are going to go to work just trying to find some synthetic accessible routes to synthesize these this molecule. And you can organize the routes up here as well. And what that does is it just sorts them by synthetic success and diversity. You'll see again, there's only 11 routes to go through here. So we're not delivering you hundreds of routes to have to sort through. And if you scroll down, again, this guy's directly purchasable from Enamine if you like ChemSpace or all different vendors here. But then you can also see there's a couple of different one-step routes which are suggested. And really interestingly, this first guy up here is actually the last step in this 2-step approach that the chemists were using. So we actually do suggest that exact synthetic strategy that the chemists were presenting. And again, we probably didn't see this in the last one, but you do have all of these exact search matches now. So you can obviously purchase this simple building block as well as look it up in the patent literature. And so that was just a simple little like literature toy example. I really want to use a more complex molecule as the last case study here to dive into some of the deeper synthetic routes that Manifold can suggest to you. So again, just going to go back to Manifold here, and over on the right, I've got a Smiles. And we were reading through some DEL literature to do some research, and we came across this one molecule here, which is a pretty big DEL target. And we really thought that it would be a good example to illustrate some of the deeper synthetic routes that Manifold can present. So I'm going to go ahead and query up this search. And so again, when you first search on Manifold, our retrosynthetic algorithms are going to work to find some synthetically success and diverse routes. So we're going to load up there. And just while we're loading, I'll pop over to the exact search tab quick, and I'll illustrate to you one of the patent references, or one of the patent databases references for this target molecule. So again, if you just go ahead and click on that, this will bring you right to PubChem to show you this molecule. And if there is any literature references down at the bottom here, I won't dig into it too much, but you could probably dig deeper if you're interested in later. So I'll come back to Manifold, and our synthesis is has returned 14 routes so far, and then it completed there. So again, just going to organize the routes by synthetic success and diversity. And you can see that there's 14 routes which were suggested, and we have a combination of like 4 steps and 3-step routes. So nothing crazy deep, but for this large, probably later stage molecule, there are some deeper synthetic routes to synthesize this guy. You'll also notice that some of these routes or almost all of these routes are really taking advantage of some really complex building blocks, which we can use these, and we can suggest routes like these because of our updated or readily updated access for the virtual chemical space. We can really suggest routes back to these building blocks, which don't need to be synthesized, can be purchased right off the shelf. And so the thing that I've been teasing around, which I haven't dove into too much, are this explore route button here. So there's a whole nother landing page that you'll be brought to if you click on this guy here, which really just outlines all of the synthetic routes in a tree data structure so that you can really traverse through and explore the specific reactions. And one thing you'll notice are these different colored bonds. And so as Aaron was mentioning, we really try hard to deliver you diverse routes, not only by the building blocks, the different reactions, but the different bonds that are being made. So we don't want to deliver to you 15 different routes with the same disconnections. We'll present to you different strategies so that you can really have an informed, educated opinion, or really informed, educated outcome from searching a manifold to the different options you have for synthesizing these complex targets. And the other thing that I'll mention here is all of these disconnections are correlated to the reaction names by color. So if you click on any one of these reaction names, what you'll see is just the, that our similarity algorithms will go ahead and fetch from the patent literature similar references based on that specific reaction which is being suggested. So again, for this amidation reaction, we suggest 14 different patent references. And again, you can click through any of these patent names and it will bring you to that specific reference where this reaction was undertaken previously. So two last things quick that I'll point out. One is this export route button. So we've really worked hard at bringing you a visualization tool for these, for exploring routes. It's not something which is that easy to kind of read through. It's easier to see it visually and kind of explore dynamically the route and the specific reaction steps to produce your target molecule. But if you really would like to kind of take it to the next step and you wanted to synthesize this route, you wanted a CRO to go ahead and synthesize this molecule using this proposed synthetic strategy, you can go ahead and export the route here. And what we've done is we've presented you this like nested representation of the route. So you have the reactions nested within each other. And most importantly, here we present to you the vendor ID numbers. So you can go ahead and purchase those building blocks, which each one of our routes disconnects back to all of these purchasable building blocks. So all the guys in blue, you can always purchase those. And then you can go ahead and try and explore the synthetic strategy in the lab or have a CRO. And the last thing I'll show is this custom route button here. And so we, again, deliver you here 14 different synthetic routes and synthetic strategies for synthesizing this molecule, all using the vast virtual chemical space. However, if you wanted to kind of go old school and design your own strategy yourself, because we didn't maybe suggest to you something which you had in mind, you can dynamically search through the virtual chemical space using this tool. And so what I mean by that is if you go ahead and you draw, you make a disconnection here. So let's say we wanted to make this amidation bond, or we wanted to break this bond to make this target. You can go ahead and use this tool to kind of craft your own retrosynthesis tree. And what we will do is we will auto-populate any purchasable building blocks with the vendor IDs. And again, you can click through any of these vendor IDs here, and it will bring you to that website there so that you can purchase those building blocks. And with that being said, I will go back to the Manifold landing page here just to emphasize to you this feedback button that we have down here. We really appreciate and hope that you guys all go make a trial account where, again, you get 30 days free, all of these features which I just presented to you. But if you have any future feature requests or problems or issues, please reach out to us on this forum. We really appreciate it. We'll get back to you guys. And I think with that, we'll have some Q&A at the end, but I'll pass it back over to Yurii, I think, for his part of the webinar.
Yeah, thank you, Emily. Hello, everyone. Thank you for the possibility to present today. I'm gonna talking about our approach to access probably the largest chemical space of make-on-demand compounds. I hope you can see my screen now. So as you know, chemical space is huge. So it's been estimated that we have over 10 to the power of 63 molecules. Unfortunately, not all of them are actually available for the researchers. In terms of the commercial availability, we're talking about maybe 8 million of in-stock items and billions of virtual compounds. And this second type of compounds has been brought more attention in the past few years because there are many cases have been published with successful results of heat identification and further synthesis of lead compounds using virtual compounds. And this is just one of the examples. Indeed, these virtual compounds are very good alternative to overused stock collections. The problem with virtual compounds is actually chemistry, because before the molecules are in the lab for wet screening, they need to be synthesized. And in many cases, reagents or building blocks to make these molecules are actually synthetically unavailable and needs to be synthesized prior to the synthesis of the target molecules, as well as the synthetic procedures have to be developed. And all this unfortunately resulted in low deliverability of the molecules from the virtual databases. And obviously this made users of virtual libraries unhappy. And the virtual compounds were seen as pollutants to the databases, and people didn't trust them in these compounds. The problem with kind of these virtual compounds is that most of them are actually generated by non-experts. And to deliver the compounds to the web, to the lab for the web testing, you indeed need to have the expertise and synthetic expertise to be behind these databases. And this is actually Pavel Mihalyuk. He's CSO of Enamine. He has published over 130 papers, and he's expert in organic and medicinal chemistry. And there are many people at Enamine who are experts in the modern techniques, and the company has generated over 3 million compounds. And obviously this database of the compounds has allowed to create a tremendous chemical knowledge. And this chemical knowledge is further can be used to generate the make-on-demand analogs of the firstly synthesized in-stock compounds. The way it works is the following. Imagine a chemist has synthesized this aldehyde in a decent amount in just 5 steps. He or she is known, and he or she used a pretty common starting reagents, and then they can repeat this procedure by again applying the same steps and just use different starting reagents available from the warehouse. So with that, they can go and generate a set of make-on-demand analogs of this firstly synthesized aldehyde. And this, again, these analogs are based on the validated synthetic procedures applied to the in-stock reagents. So this is what we call a make-on-demand concept. And because the reagents are known and the price or the workforce that is necessary can be calculated, this make-on-demand analogs are always come with price and estimated lead times, which basically creates not just a database of the molecules, but the catalog. So these make-on-demand compounds are actually something in between the in-stock items and the custom synthesis, where anything is not known and you need to start building the synthetic road from the scratch. There are different make-on-demand products available. Those are make-on-demand building blocks or made building blocks, real database, and the real space. And that's actually something that is coming soon, the updated real space of over 17 billion molecules. I want to talk a little bit about this made building blocks and how have they actually created. As I said, the building blocks are typically molecules delivered on a gram scale, And with application of the flask chemistry, so just the typical chemistry we used to do in the labs, and the building blocks are synthesized from the in-stock reagents in up to 5 synthetic steps. This is a publication that is describing this make-on-demand building blocks concept that's been published pretty while. And just few things about these building blocks. They're actually not the reagents. They're rather advanced reagents that you can use to optimize your molecule. Currently, the database of made building blocks contains over 282 million molecules with up to 20 effective heavy atoms. And it includes all the necessary classes that medicinal chemists need in their projects. And those are just a few of them. You can search for made building blocks using ChemSpace web page, where you can have all the necessary options like substructure, similarity, exact match, and the search is pretty fast. It's enabled thanks to Nextmove software. Few words about real compounds. So unlike the made building blocks, these real compounds are actually compounds delivered by one-pot parallel chemistry, and they are ready for synthesis and ready to be included in any assay. We use the compounds that are only up to 3 synthetic steps from the starting reagents, and the compounds are typically synthesized on the milligram scale. This is the recent publication that shows basically the generation and the expansion of the real space. As I said, we have billions of real compounds to test. Current version of the real space is based on the 184 validated synthetic procedures, applying a simple chemistry like this one, or the advanced synthetic procedures. I would like to mention that almost all the procedures are actually published and shared with the synthetic community. With the new version of the real space, we're actually introducing obviously new reactions and new building blocks which allow the space to grow. And the current one contains over 125 building blocks, in-stock building blocks that are used to build the space. We do not include all the building blocks, but only the qualified reagents. That means that we analyze each reagent experimentally. For example, this reaction, we tested this aldehyde here, and obviously we set not one experiment but several. And if we see that the aldehyde behaves well in this synthetic procedure, we basically include this molecule in the enumeration. For example, this aldehyde showed rather not very good success rate in this procedure, so we decided not to include it in the enumeration using this procedure. But what we're trying to do, we're trying to fight for each building block, and if we see that this reagent is not compatible with the conditions of one procedure, we always try to find the alternative procedure to basically include it in the enumeration. Yeah, and these are just few examples of over 5,900 new building blocks included in the new real space. Currently, we have few options to serve the real space. One is the conventional database, which we call real database, which has 1.95 billion molecules. And we have the real space, which is basically enabled by F3 search. The database and the space, they're overlapped, and the database is rather a subset of the space with a focus on on a small and more interesting part of the space. This is just a few numbers related to the database. So it's the molecules from the database comply with a rule of 5. You can see that many of the molecules are in the lead-like chemical space, and we do not have any paints or other toxic motifs in the molecules. The database, the real database, is searchable on Enamine store or on chemspace.com, or it's searchable via the KNIME nodes. There are a few examples published to date using the applicability of the real compounds, and again, just a few of them I would like to list here. Now I would like to talk a little bit about the exploration of the real space with the F3 based similarity search, and just to give you a very high-level explanation of what that is. It uses the F3 technology developed by Buvailo IT, and what it does, it actually searches not in the classical structural space, but it uses the so-called fuzzy similarity that includes some features of the molecules. So each molecule is split into these nodes, you can see, and each node has its set of features. And when the search runs, it basically compares the features of the query molecule to the features of the molecules from the database. And after the search is done, you can see that it basically generates this again fuzzy similarity, which is not the classical Tanimoto similarity we used to use. And if you just see those molecules and analyze them, you can see that they have some similar fragments in their structure. And the molecules can be mapped just for easy visualization and analysis. The strength of F3's enabled search is that you can do the scaffold hopping. So basically find molecules that are dissimilar in terms of the classical structural similarity, but might be similar in terms of the features. And this is a paper that we published together with Buvailo IT, which basically demonstrates the application of this approach to the search in the real space. You can try F3's enabled search in the real space on chemspace.com, or you can try the Infinity tool provided by Buvailo IT. So chemical space is really big, and we are just exploring a very small portion of the space. We're trying to expand that dot, and we do our best to make it bigger. With that, I would like to thank you to Andrii and all organizers, and like to thank you to Enamine, ChemSpace, Buvailo IT, Next Move, all collaborators from UCSF, Harvard Medical School, and CMU. And thank you to everyone who trusts in the Make on Demand.
Yeah. Thank you very much for all the presenters for providing such valuable insights. At least I was kind of entertained. And as you probably have observed from the number of talks that we just went through, that the powerful medicinal chemistry is where chemical technologies are combined with the latest computational technologies like AI. And basically, artificial intelligence is a very hype thing right now. And a lot of guys already know how powerful the tech is, but others are still skeptical. So I've been on the conferences where I've seen some debates on this topic, because a lot of computational chemists, for example, they know that the AI is fairly old concept that originated in the 1950s of the last century. So essentially, but what I have to say that the pharmaceutical industry is now going through transformation, a major transformation driven by the data technologies and AI. And so to conclude this webinar, I'd like to present a little overview about the latest trends and where these AI technologies already changing, already, I would say, revolutionizing the drug design and chemical synthesis. Well, let's start first of all with what is this thing AI, artificial intelligence? Why is everybody talking about it? In fact, it's a lot of confusion in the industry regarding the terms. And when everybody is talking about AI, in fact, in most cases, they're talking about machine learning, some kind of machine learning things, or more specifically, deep learning algorithms that are truly shined over the last like decade or something like that. So the concept of AI is indeed a quite old concept. And the first program that was playing checkers was invented, I guess, in the 1950s. And since then, a lot of revolutionary breakthroughs have been done in this area. But the problem is that the problem was in the last century that the computational power was not there yet for the AI to actually show its power. And basically, the computers of the past were not able to provide that level of compute power to perform very data hungry manipulations, algorithms, predictions, etc. So that was pretty much theory at that point. But with the emergence of specialized hardware like GPUs, powerful computers, and cloud infrastructure, the old concepts, they shined. They basically were reinvented in new forms and ways. And now the time has come for the truly transformative, like some refer data-driven revolution. So, and basically the technology is peaking right now, and it was primarily driven by the discovery, by the major development in 2012 when the very famous ImageNet competition took place, where the deep learning algorithm showed extraordinary predictions of the images, basically being able to recognize objects on the images. Now nobody is, it's like common practice that AI can recognize images. Now nobody is excited about this anymore because this is already very, common technology. When you go to Facebook, there is a feature that basically recognizes faces on the photos and basically can assign people to the faces. And in fact, this is tremendously complex task, which was impossible some 20 years ago. It was virtually impossible. Now it is a very commercial thing, it's a lot of open source software that can do that thing. This is the reason, this tremendous progress is the reason why pharma already picking up this technology as well. And in fact, a major shift was done in generative chemistry, for example, in 2015, when the concept of generative adversarial network was developed, when there is, in a very general terms, of just two neural nets, the one which is trying to sort of mimic the prediction, and the other is trying to kind of prove it wrong, and they are kind of competing automatically together, learning from together, and basically going step by step to the ideal result. And these generative neural nets were very effectively applied for generating chemical compounds with predicted and wanted qualities, like, for example, drug-like chemicals, etc. A lot of discoveries have been made in the heat discovery space around 2018, 2019, where really like tens of companies reported different advances where they managed to predict hits to some known or less known biological targets, and the hits were either generated or found over the screening campaigns, and they were tested in vivo and showed efficacy. And things are really moving very fast in this area. But you probably want to ask, how is it different from good old chemoinformatics or, you know, statistical methods, linear regression, which was used like 50 years ago probably? And the point is, the AI is not an algorithm. AI is a system, and the revolution that is now everyone is now talking about is not because somebody, you know, tried a new algorithm and applied a new software package to a certain problem, but because some companies managed to build systems basically where the process is closed in a loop, and this loop allows for the learning process to occur when the data is fed into the system, and then experiment runs, the prediction runs, then experiment runs to prove the prediction either wrong or right, and then the data about either failure or success is fed back into the system, and then AI learns, and this whole loop is basically automatically back propagated. So some companies did manage to build this kind of systems, and in fact most of the big pharma companies are now implementing programs internally or partnering with companies like PostEra, for example, to basically probe the ground in this groundbreaking technology. As of now, AI is applied in virtually every step of the drug discovery process. Here is a tree. I have written a tree where you can see that it's basically starts with finding materials, consumables, like and doing some chemical synthesis prediction, and this was the topic of some of the talks today. And then data mining from literature, building knowledge graphs, basically building ontologies of diseases and biological models of cells, etc. Then assisting in screening programs, finding hits, lead optimization, and etc. Needless to say, the lab experiment is of course has to be conducted. Still, AI is not at that level that all the prediction can be run all the way to clinical candidate. No, it's not the case yet, but now already a lot of time, energy, and cost can be saved by applying these technologies. So let's just briefly go through some examples. Let's start with very obvious thing. There are a number of computational marketplaces right now which allow a very powerful search to be run on reagents to find, let's say, antibodies or molecules or different other chemicals or biological substances, and basically create the experiment in a very powerful way. It's not like search, and very usual search. It's not like, you know, going to a catalog and basically trying to find something on the catalog, but it is where the you know, the AI system is learning previous searches, and basically every next search is becoming a little bit more accurate than before. That's the power of this technology. And for example, there is the BenchSci company which has received funding from Google to develop the biological search system. It's tremendously powerful. And also there is the chemical synthesis planning spaces is now booming with AI technologies. There is some programs like, for example, Cynthia, which was acquired by Merck some time ago. There was a very well-known program, Chematica, at that time. And the Manifold, of course, which Aaron and Alpha presented today, and another example, and IBM Ericsson for chemistry, which is an open source synthesis prediction program. So, I mean, it's kind of a lot of them right now on the market, and they're very powerful, as you have seen in the presentation before. But the next step in this, remember, I told you that the AI is not, is not a program. AI is a system. That's the power. That's where the industry is heading. And so the next step is to build in this system, which would do all the steps automatically without any human intervention or with minimal human intervention. Where, for example, the process is done the way that the target compound basically is predicted by the algorithm, then the synthesis predicted for that compound, and that synthesis is basically the roads are sent into a certain format to the virtual laboratory where robots basically mix components and do some chemistry and report results like NMR or some measurements directly to the laboratory. This whole process is not a theory, it's not a sci-fi, it's actually a thing that is already existing, and some companies do that, and even offering commercial infrastructures for running virtual experiments from your home. You can program the experiment from your home. Now they have a lot of limitations at this moment, but again, the progress is very quickly progressing. So, for example, this system was developed at MIT in collaboration with Bristol Myers Squibb and other companies. And there is another example is RobRxN by IBM, which is also the cloud-based laboratory where you can run chemical experiments. Another area which is a very powerful use case for the AI application is biological target discovery and validation. Essentially, this is where the big data comes to play. You see, there are millions of reports, research papers that are published basically on a yearly basis, and no one chemist or biologist can track everything. And even if you try to search using your usual browser, Google search or anything like that, you're not going to cover a lot of results because the search is not perfect, and you can't go through hundreds of suggestions that the search would suggest you. But the AI systems, they now can basically parse the internet and read the data through the natural language processing algorithms. And they can recognize concepts like, for example, a protein. The AI can extract data from the paper, and it can and understand that this one term, it means the drug, or that one term means a chemical, and that one other term means a biological target or pathway, etc. So the AI does it extremely fast, like on millisecond basis, and then the algorithms can connect different concepts into so-called ontologies. Ontologies essentially graphs of knowledge where different entities are connected. Let's say you have 1,000 papers on a specific cancer type. Then the AI can basically go through the papers and analyze the information and build the model of that specific cancer type by extracting knowledge, experimental knowledge, and connecting the dots, essentially building the ontology of that cancer type. And then based on that, it can suggest certain targets which might be relevant for curing that specific cancer type. And then of course, that needs a validation. And this is where experimental testing has to occur in the lab, and results either confirmed or declined. And and again, the system is learning and it's improving on itself. The next logical step, and this is also a very powerful topic for and use case for the AI application, is heat discovery. Essentially, there are several strategies for heat discovery, one being a docking-like experiments where essentially there is a known structure of the biological target, which might have been predicted on the previous step, by the way. And and the AI is trying to optimize the docking functions or somehow create molecule from scratch, for example, and feed it into certain constraints of parameters that are needed for that specific biological target. Another major strategy is generative chemistry, where essentially the molecules are imagined, so to speak, and they imagine in a way that they are already possessing all the specific parameters like drug likeness, specific molecular parameters, etc. And then all that we need to do is just test and see if the prediction is right or wrong. And believe me, from analyzing a lot of literature, they are getting really, really powerful at that. So next, the preclinical research. What follows the study of successful heat discovery and lead optimization is the preclinical research, and AI is shining here as well. There are a number of companies who offer already today the computational remote labs where you can basically set up the experimentation. For example, a massive assay screening experiment where you basically send the parameters, you basically set up the experiment, and the lab with the partner is running the experiment, reporting results, and the AI can kind of optimize the conditions of that experiment based on real-time observations. And again, it's learning and it's getting better with each major program, with each major iteration. And finally, as I said again and again, the AI is not a program. AI is not a Python library which you can import into your software package. AI is a system. And the system is something that gets data, does something to it, processes results, and gets meaningful output into the world. That's the AI system. That's where real power of AI lies. And there are a number of companies who actually managed to build this kind of systems, where essentially they start with literature search and they end up with a preclinical drug candidate, which needs to be then tested in vivo, in vitro, in vivo, and then basically processed, selected for further clinical trial programs, etc. There are just a couple of companies that are already doing that, like in silico medicine, Hilks, and others that are claiming that they created the whole system, which covers the whole spectrum of drug discovery. But they have also some issues at the moment, and of course the progress is still ongoing, and a lot of research needs to be done until we can truly do pharmaceutical discovery in silico. With that, I'd like to conclude this little introduction and overview about the AI trends. And I'd like to welcome all of you to Clubhouse. This is a new, and very hypey social media where people can talk like on the radio podcast. It's very entertaining and very funny. So just follow Enamine there to be notified about our upcoming webinars. We are planning a webinar in April. On a very exciting topic. The topic will be announced a little bit later through our social media channel. So follow us, follow PostEra. And also we are planning a little Q&A session about the things, about the manifold, about chemistry, about life in general. And we are planning it to host tomorrow, February 24, actually the time will be probably a little later, but follow Enamine to be notified about this event. So with that, I'd like to thank you everyone for joining our webinar. Please visit Enamine website and PostEra website to learn more about specific products. And I'd like to thank you everyone for joining us, and our speakers for great presentations. Thank you, and with that we are concluding.
I believe there's a few Q&A questions. I think that got missed off at the end. Shall we try and just run through a few of them, Andrew, while a few people are still around? Okay, I'm going to take the silence as a yes, and I'm just going to dive into the Q&A. I think we may have lost Enamine here. So thank you to those who sent in some questions. Let me try and at least answer the ones that pertain to PostEra. So let's start up from the top here. Actually, I guess chronologically, Alfred, do you want to start out with that first one, if that's okay, and just unmute?
So yep, Alpha here. So I think thanks Ernst for asking that question. I think we focus on non-covalent inhibitors because they're usually known to have less idiosyncratic toxicity down the line, and the fact that we found such a potent molecule being non-covalent is a great impetus for us to continue this pathway. Obviously, if we find that the molecule, needs to be more potent, which actually, seeing the current data that we have, it seems that we are very close to candidate already, then we can always add in and warhead later on at the end. And I think your point about MPRO being distinct to human homologs is a great observation. That's exactly why we think we are seeing really good protease activity, and that's why I think it's a great druggable target for COVID.
Thanks, Alpha. I guess I'm going to take the next one. So thanks to the individual who asked about Manifold. So I'm just going to read it out because I believe a few people can't actually see the question. So the question is, what about intellectual property regarding Manifold? How is exclusivity and confidentiality of compounds treated? Are the query structures treated in terms of confidentiality? The answer is very simple here. Obviously, if you search in Manifold and you find a purchasable compound, well, there's no IP to be over that compound given it's in the public domain. However, if you enter a target compound that Manifold designs a synthesis route for, you will most certainly own the IP over that target structure. PostEra does not make any claims over that. And when you come to order those compounds, we have agreements with all of our vendors and CROs in terms of the level of confidentiality that you would like for those when those compounds are being made. So hope that covers that question. I guess, Emily, maybe I set it up for yourself here on the Mirko scaffold.
Sure, yeah, I can. This last question, I guess this next question, Serena asked, what is the difference between the two Mirko substructure searches exactly? So essentially just the idea is there in the query molecule, you have either, or you have both a Mirko scaffold and an extended Mirko scaffold, where the extended Mirko scaffold also accounts for the location of the substituent or of the substituents on the scaffold. So those are the two differences for the matches there.
Can it be, sorry, Emily, can it be like a Bemis-Mirko and Bemis-Mirko-Luz framework with the heteroatoms assigned? So maybe is that the case of
Do you want to explain on that one a bit, Alpha?
I think the two Mirko are sort of how to exactly define a scaffold with or without heteroatom and connectivity, right? I think.
So it's just like just a scaffold without assigned heteroatoms and.
Yeah.
Okay.
Okay, I'll run through a few more here. So it's a question about whether Manifold includes virtual spaces from Enamine and Wuxi. The answer is yes, absolutely, from both. So this is specifically from Lab Network that Wuxi provides. And obviously Enamine is certainly the starting point of which Manifold was built upon. So yes, they are available through Manifold, both in similarity and synthesis, and they are continually updated. The next question is there a way to see pricing from the different vendors, again in Manifold? So the answer here is that pricing in terms of visibility will be coming at some point very shortly. And just to add two caveats, the reason why we didn't rush so quickly to display pricing was twofold. So firstly, particularly if you're ordering singleton compounds, much of the all-in cost of that order can actually come from the formatting and the shipping. And so ultimately, I don't want to call it misleading, it's not misleading, but it doesn't ultimately give you the full picture of the compound, and really only the formatting and the shipping can come when you've actually put an order into the vendor. The second thing that I would say is sometimes catalogs can be outdated, so it's always good to check by speaking to the actual supplier whether that price is still valid. Like PostEra works incredibly hard to make sure all of the databases are updated as frequently as possible, but there are exceptions, and so just it's worth being aware of that. And then I think the final question, unless people in the audience want to just jump in and ask anything to the team here, the final question is, what is the difference between Manifold compared to SciFinder and Reaxys, as they also provide commercial sources for compounds? This is a great question. We are very aware of SciFinder and Reaxys, and we've used those tools and are a great admirer of some of their capabilities. The one thing we can definitively say is that these tools do not have the huge underlying virtual spaces, certainly not dynamically updated like Manifold is giving you. Practically what that means is sometimes 3 or 4 step syntheses are actually just 1 and 2 step syntheses, because of the existence of complex functionalized building blocks that are now increasingly ubiquitous. And additionally, I would also say that I'd like to argue the way that we present retrosynthesis is a little clearer. We often find that particularly believe it's Reaxys generates a lot of routes that you then have to try and work through to figure out the most viable ones. So we do think there are some competitive advantages there when it comes to the retrosynthesis. I believe a new question just got shot in, so let me read this out. Similar to COVID Moonshot, can we expect an increased demand of shared computing power to run molecular simulations on other medical projects in the future? So I believe this person is a folding at home user. I can't comment specifically on this, but all I think I can definitely say is that we hope COVID Moonshot has provided a really great precedent for people to see that crowdsourcing both chemistry and even compute is possible and it is doable. So if you speak to the Folding at Home team, John Chodero, who's one of the core leaders of COVID Moonshot, actively runs the work at Folding at Home. He'd be delighted to talk to you about the future plans that Folding at Home have.
Yes,
I think
I would like,
sorry.
Yes, on this I would like to add that also another scientific group at University of British Columbia from prostate cancer, they are also working on creating of open source docking platform just for free docking.
It looks like we have another question that came in. I think only the panelists can see this one, perhaps. They were asking about, are there any plans for us to implement enumeration of large searches? For example, returning hundreds of thousands to millions of compounds with reaction trees over a wide search for like future refinement and filtering with their in-house metrics. So the answer to that is, so this is essentially API access to our retrosynthesis. And we are planning some API access to be released shortly. Largely that will be focused on the exact similarity substructure searches. However, the synthetic routes, we probably won't be delivering that. However, some other metrics such as like a synthetic accessibility score will potentially be delivered in that API.
Great. Was there any more questions from the audience? I know we've covered a lot of ground, so we can wrap it up unless there's any final questions.
Yep. So again, thank you very much, everyone. I guess we can conclude now, right?
Yeah. Thanks.
Yep. So it was a lot of fun. Yep.
Yes, bye.
Bye.
Thank you.
Bye-bye.